Okay, welcome everybody. We've got half of chapter 15 left to go through. I mentioned Monday. We're going to cover this today. We won't have class on Friday. The exam will open up Friday at 9, and you'll have the opportunity, if you feel like it, to take it during lecture time. It is like the others. It doesn't close until Monday at 5 p.m. So you still have the same window of time. Any questions about that stuff before I jump into the details? Okay. So the last bit of this chapter, we want to think about a handful of different mechanisms that microbes, bacteria for the most part, as we're moving through here, but different things that microbes potentially can do or may utilize to truly gain hold and establish disease, and some of the potentials for that. We will go into greater details when we're talking the individual organisms a little later on. But we'll see a few mechanisms this morning as we go through this stuff. So in this first example, we've got a patient with some edema, some pretty good swelling in their hand. And there's lots of things that potentially could be causing this. Maybe, as our example here, some bacteria that are stimulating this inflammatory response. And there's lots of potentials when we think about inflammation, and oftentimes think about it as a bad situation. Maybe, altering our mobility or something like that, a sprained ankle that's very inflamed. We may try to do something about it, take some anti-inflammatories and try to bring the swelling down. Try to minimize the symptoms that we are experiencing with that. And oftentimes, that seems beneficial to us. But sometimes when we think about the inflammatory response, really our body is trying to help combat whatever it might be. And we've got our example here, this bacteria that's causing some sort of a reaction. And in turn, our body, and we will touch on this in the next chunk of material, lots of different ideas when we think about immune responses. But a lot of times when we initiate any sort of a response, we see some inflammation that's kind of following right along with it. And the idea would be to really help, like, call in the troops to get immune cells, to get proteins in the fluid that's moving in, like antibodies, try to get substances to the location wherever we've got this problem as rapidly as possible. And there are lots of immune cells that are traveling through our circulation, and in an inflammatory response, the vessels become more loose and lend to these immune cells being able to wiggle through these cells and get into the tissues to ultimately try to try to combat whatever it is that's causing these problems. So some of the responses stimulate, there's all sorts of different molecules, some stimulating a pro-inflammatory response. Other molecules being released might be stimulating different types of immune cells to come in and do some mopping up, if you will. So lots of stuff within this fluid that's causing the edema that's potentially really trying to be a benefit in this situation. So there are a lot of different potentials when we think about organisms trying to get in and gain hold, or to migrate or move through the body. Not all of them harbor the properties that we're going to be talking about, but some of them do. And when we think about some of these examples, really the idea with this would be a virulence factor, enhancing the potential to cause disease or make it better at doing its job of eliciting this disease state in us or something else. All right. So we got a couple of examples here, a normal situation and then a bacterial example that gives these cells, these bacterial cells, opportunities to get into our own cells. So hyaluronin is a polymer that's between the epidermal cells, connecting one cell to the next, kind of like sort of like a cellular glue, and helping to ensure that they're all kind of together. All right. Like I said, some, not all, but some bacteria may harbor some potentials. The enzyme hyaluronidase, and typically enzymes ending in ases, right? So the enzyme hyaluronidase acts upon hyaluronin, all right? And it basically helps to dissolve or break away that glue, that cellular glue, and allows the bacteria the capability of gaining access to the insides of cells as it moves through there and destroys those connections. So that's the idea, giving them the opportunity to move through and penetrate that, lending to the potentials for disease, whatever it might be, to really proliferate, and the bacteria gaining access to deeper tissues or potentially into the circulation and lending to a septic situation. There are all sorts of different microbes, different bacteria causing disease. We've got a handful of Clostridium species that we're going to think about today. They truly hold some serious mechanisms to cause issues, to cause harm within us. All right. And there are a boatload of different Clostridial species. The potentials for issues really depend on who we're talking about as far as what sorts of stuff they might do. All right. So we've mentioned briefly Clostridium difficile. We talked a little bit about it with some intestinal issues. We talked a little bit about perfringens that we've got right here. And we think about some opportunities with it, with like gas gangrene, flesh-eating sorts of ideas when we think about some of these guys, all right? So this mechanism really lends to that capacity, all right? It's not the same, it's not hyaluronidase, it's collagenase, same but different, all right? The collagenase allows the bacteria to degrade the collagen and lending to it gaining access to, either into the circulation, or we could also think about into tissues as we're moving through, thinking gangrenous sort of situation. So here we've got a vessel, the endothelial cells, and the collagen surrounding this as it breaks it down and then gains access into the circulation, into the lumen of the vessel, lending to a septic situation. And that's what we're looking at here with this actual image. So it allows the bacteria truly a potential to cause some serious damage as we think about that, and lending to it, once it's in the circulation, to cause greater disease. So this image is a little snapshot of a gram-negative bacterium, all right? And there are some details with the positives or negatives with different potentials for those cells to initiate disease responses or to stimulate an immune response, either way, some of it based off of cell wall characteristics. And with our gram negatives, they're a little bit different than the positives with the thin peptidoglycan layer and the outer membrane. The outer membrane that becomes important when we think about some of the disease process. And that's really what we're looking at with this, right? This is a little bit of that outer membrane that is what really causes some of these issues. So there's some lipid A and O antigen that are components of the outer membrane, this lipopolysaccharide layer of gram-negative bacteria, right? And the lipid A and O antigens together stimulate some of these ill effects. The lipid A is a promoter of inflammatory responses and fevers, and that in turn stimulates a lot of the symptoms that we're seeing with this. The O antigen could stimulate more of an immune response. As we think about humoral and antibody-mediated response with those antibodies being able to recognize. There are all sorts of different potentials when we think about trying to fight bacteria, and we've talked about some antibiotics, and we talked a little bit about modes of action. There are antibiotics that are narrow spectrum that target specific types, and those that are more broad spectrum that are more generalized antibiotics targeting groups or positives and negatives alike. And there are different types of infections where we may see maybe warranting the use of an antibiotic or maybe not. And in some issues, kind of lots of issues, with some GI infections, oftentimes they're gram-negative organisms that are causing the gastrointestinal conditions. Our Clostridium difficile is an exception. It's a gram-positive, all right? But lots of gram-negatives can cause some problems. Bad E. coli, Salmonella, Shigella, all of those gram-negative bacteria, and treatment with some antibiotics ultimately can cause the destruction of the cell, releasing more lipid A that in turn is a toxin, right? And that in turn can cause greater issues of inflammatory and fever states, maybe lending to more of a serious condition. So it truly kind of depends on some of the circumstances and who that culprit really might be as far as warranting the use or not of a specific antimicrobial. So there are other ideas of, as we move through here, that might be like how molecules or toxins or microbes themselves might be able to gain access into a host cell. And in some circumstances, a toxin, in this case, is able to make contact to recognize a specific cell based off of some receptors. And if that cell has the receptor on it, the toxin can link up and bind, and then it stimulates the cell, it stimulates it to bring it to the inside through endocytosis, through it basically pulling it to the inside. And it does that, as we see in this middle frame, through forming a little vesicle, a vacuole that basically pinches it off to the inside. Now we've got a membrane-bound vesicle inside of it, all right, inside of this cell. So the idea with this, we've got a multi-subunit toxin. And really the business end of it is the top bit, the A subunit. It's the one that's going to really be causing the problems. We'll see the details of this in the next slide, all right? This is just initially how do we get it in, all right? So the B subunit links up to that receptor, basically allowing it to dock. We pull it in, and the A portion gains access to the cytoplasm, to the inside of the cell. All right. So as it moves in, it separates and makes its way where it truly wants to be. All right. So an example of this can be some toxins from diphtheria. And we can see this mechanism here as we are moving into the cytoplasm and gaining access. This toxin from diphtheria basically causes the demise of that cell. It interacts with the protein synthesis machinery, basically driving a wedge in the synthesis of proteins. So it blocks this activity. Our elongation factor, which should be lending to a growing protein chain, it interacts with the elongation factor and basically stops this process. So it transfers an ADP ribose, as we see here, our NAD, nicotinamide adenine dinucleotide. We transfer this to the elongation factor, and it basically alters its shape, and it causes it to detach from the ribosome, basically halting the synthesis, the growing polypeptide chain. And that ultimately, once we start blocking all of the ribosomal activity, drives a wedge in the cellular processes, and that cell dies. That's a mechanism that diphtheria utilizes in part of its progression of disease. So I told you a minute ago that we've got a handful of Clostridium organisms to think about. Here's two more, all right? Botulism, Clostridium botulinum, and tetanus from Clostridium tetani are almost like the opposites of one another as far as what they do, all right? They both impact nerves, but they do it differently, all right? And it almost makes you wonder if you couldn't maybe counteract one with the other. I don't know if that's a potential or not, but it sure seems like something that should be evaluated. So the botulinum toxin, we'll start with it. It's on the top. So it causes a flaccid paralysis. The muscles stop contracting, and they just can't because of this basically blocking this activity. So we've got two examples, the normal and then the abnormal. So when it's functioning properly, and then the activity with the botulinum toxin causing the abnormal situation, all right? So our normal situation, we've got our nerve right here, and then our muscle cell down below with receptors on the muscle cell. Our nerve with this acetylcholine is going to be dumping acetylcholine, linking up to these receptors and stimulating the muscle to contract, all right? That's the idea, all right? If it's functioning properly, all right? So a botulinum toxin blocks the nerve and blocks the release of the acetylcholine, so it can't release the acetylcholine. It doesn't link up to the receptors. The muscle doesn't contract, all right? They use very low levels of the botulinum toxin for Botox injections, and it does this mechanism, but on a smaller scale, right? On a localized scale, where the injections went into your face, all right? Not on a systemic scale where it's happening throughout your body, all right? That's a big concern, all right? And botulinum toxin through foodborne illnesses or other situations, typically thinking food, causes a systemic situation, not just a localized, expressionless face, right? So it truly kind of depends on your circumstances, right? So stopping the contraction, flaccid paralysis, right? The tetanus is the opposite, right? This is spastic paralysis. Muscles contract and then don't relax, right? That's basically the issue with this, right? And there are handfuls of images that show some of these examples. Sometimes we talk about tetanus and say lockjaw, where basically your face is locked in this kind of smile, kind of creepy. Makes me think a little bit like the Joker, a little bit with that as you see this, and you just can't stop, right? There's other images with individuals that look like they're bent the wrong way, with their head almost touching their feet as they're bent around backward. They shouldn't bend like that, but they can't. They can't help not, right? Almost seeming like maybe some sort of exorcism could be warranted with that situation, right? As you see them bent backward like that. Seems very unnatural, right? So the extreme opposite of what's happening with the botulinum. So in the normal circumstance, we've got mechanisms to basically stop the contraction, right? So, and we're not showing it up here because we don't need to. So the glycine and GABA, they link up to this axon and basically stop the release of the acetylcholine, and the muscle is able to relax, right? So turning off what we're seeing up above in this normal situation, right? Shutting it off. Now we go back to the relaxed state. And in this circumstance, we block that activity. So we prevent the release of the GABA and the glycine. That causes the axon to just continuously dump acetylcholine, and the muscle stays in this spasmodic contraction. Can't relax, right? So, and this truly a serious situation with, you know, these toxins causing in either circumstance these issues, and it really kind of just depends on circumstances. We think about, you know, tetanus and some vaccines against tetanus to help prevent that, and you know, something that you should be you know, kind of aware of, that you need it about every 10 years. If you step on something, you get a cut or something like that, they always blame a rusty nail, something that potentially may bring the bacterium into your body. And typically it would be the spore stage of this, because it's probably out in the soil and it's anaerobic. So you'd see a spore, you wouldn't see it, but you could see the spore on this nail just waiting for a perfect opportunity. And through a puncture wound, it gets it into your body, and in turn is able to germinate, start growing, and then start releasing these toxins. And similarly with, you know, food preservation or whatever with our botulinum. So another mechanism that we can potentially see in some bacteria is the ability for them to kind of hide or cloak themselves with a sugary coating, a capsule layer that they secrete and surround themselves with. And that can provide an opportunity for that bacteria to go undetected, all right? Immune cells or antibodies, as the surveillance is happening, they're trying to detect signatures like little antigens, proteins on the surface of these cells. And if it's secreted this sugary coating, the ability to see that stuff or to detect it is dramatically reduced. So kind of hiding that bacterium, right? And we can see here a non-encapsulated and then the encapsulated bacteria. The sugary coating provides this very, very thick barrier that hopefully prevents this activity of recognizing the cell underneath. All right. We oftentimes look at bacterial cells, and if they have capsule layers, we describe that as another virulence factor, another potential that gives it a leg up at causing disease. All right. Streptococcus pneumoniae, which I don't know if that's who we're looking at right here or not, but it could be. All right. Streptococcus pneumoniae is a gram-positive cocci streptococcus. So spherical shaped bacteria. It typically, when we look at it under the microscope, is two cells together. We call that a diplococci. And there are different types of Streptococcus pneumoniae, encapsulated forms and the non-encapsulated forms. And if you happen to have the encapsulated form, well, now we start thinking about the potential for it to truly cause pneumonia because it can go undetected. And if it's the non-encapsulated, in those circumstances, it's able to be visualized by our immune response, right? So we won't go through great detail with this, but there was some research done with Streptococcus pneumoniae, really looking at the ability for bacteria to pick up stuff from their environment. And we talked about some of the mechanisms that bacteria can utilize, right, for getting extra material into them. Conjugation, where a plasmid allows, extra material to be transferred from one bacteria to the next. Transformation is the uptake of naked DNA, and it was first looked at with Streptococcus, right? And then the transduction using a bacteriophage to inoculate that stuff, right? With the transformation, the uptake of naked DNA, they were looking at Streptococcus, and they were looking at what Streptococcus pneumoniae, and they had two different types, the non-encapsulated and the encapsulated forms. And you could take the non-encapsulated form and you could inoculate that into a mouse, and the mouse would live because its immune response could kill that bacterium. And you could inoculate the mouse with the capsule form of that bacterium, and the mouse would die because its cells couldn't recognize this pathogen. All right. So then the researcher took some of this encapsulated bacteria and he killed it with some heat, right? And then the non-encapsulated bacteria was grown up with the dead bacteria that were able to form those capsule layers. And the non-encapsulated pulled up some of that DNA from the dead capsule formers and incorporated it, and they became capsule producing ones. So they were able to show you could take a non-pathogenic type and exposing it to dead cells that had that potential to be pathogenic, they were able to pick it up and truly showing this transformation process with something like this, right? The non-encapsulated picking up the details from the dead ones of these, right? Pretty cool. So, like I said, I don't know if that's truly what we're looking at in this image or not. I don't know if the text tells you who we're looking at or not, but that's who I want it to be, because that's what I feel like it sort of looks like right here. So in this case, we've got our capsule layer that's blocking access to these antigens. This guy right here, this Y-shaped thing, is an antibody that can recognize, or should be able to recognize, these antigens. On a non-encapsulated bacteria, the access to it is easily obtainable. We can get right on there and stimulate this response. But on the capsule layer, the antibody can't link up to it, all right? So if it was able to do that, another white blood cell could recognize the antibody, and in turn, that antibody would stimulate it to link up and to destroy it, all right? And that's what would be happening to these guys up here, the non-encapsulated forms, all right? There are other potentials. Some of these bacteria that form the capsule also can be secreting substances called proteases. A protease is an enzyme that destroys proteins. There's lots of protease activity. We see lots of that in our intestine or in our gut as we're breaking down stuff to get the individual amino acids and use them for our own. So bacteria may release some protease, and in this case, it could target antibodies that may be trying to link up to the cell and destroying them before the antibody can take hold and a phagocytic cell recognizing it. So there are some other potentials besides just the capsule layer that may give them a greater opportunity to stick around, right? And maybe really to stimulate a disease process in the first place. Okay, so lastly, some viral stuff, all right? Everything that we've really looked at has been bacterial so far, but there's definitely some other opportunities. And, you know, when we really transition over and start thinking disease stuff, we'll talk about all sorts of different microbes and some of their potentials to utilize different things to get in and take hold, right? So more than just bacteria, all right? So this image is an example of some potential things that can occur with the flu. And, you know, influenza is something that is potentially life-threatening. Really kind of depends on the circumstances. Every year, people die from the flu, all right? But sometimes we see the mortality rates with the flu considerably higher than other years. And it goes with some of what's happening in this figure, all right? Kind of just depends on the circumstances, sort of a perfect storm occurring to lend to those potentials. So influenza is an RNA virus, and in turn, that opens up some potentials for greater amounts of mutations occurring than happening in a DNA virus, where there's some proofreading mechanisms in place, all right? So RNA viruses tend to change more rapidly than DNA viruses, all right? And this example here, there's two different ways that some of this change can be happening, all right? Antigenic drift is just some mutations occurring. There's all sorts of potentials with these mutations to cause some alterations, maybe making it a little more potent or maybe making it a little less potent, kind of just depends, all right? These spikes on our virus, the neuraminidase and hemagglutinin, there's all sorts of different potentials with these spikes as far as their respective sort of protein signature, all right? Subtle variations, and in turn, giving us multiple strains of influenza viruses, some of which are maybe a little more common, and others maybe not so much, and potentially greater concerns, right? Just dependent upon what you got going on, all right? So this antigenic drift, just a change occurring, and this would be, we could think about occurring in us as we are infected with this one strain of influenza, and then we get a slight mutation, and what we sneeze out to others for them to get infected with is slightly altered from what we were infected with, right? And in this case, the hemagglutinin has been mutated the littlest bit. It's changed from what we were initially infected with. This is antigenic drift, just a subtle mutation or few mutations that cause some variation over time, right? In this next case, this antigenic shift is lends to more potentials for greater change, right? So this person is infected with two different types of influenza, all right? So virus A, virus B, and we've got a little bit of different genetic material going on in both of these. We can see their H and N antigens on here, different looking, all right? And as we are co-infected with these two influenza viruses, as they go through the process of the viral cycle and replicate within our cells, as they are packaged up and released out in this burst, we can result with something that is completely different from either of the two that we were initially infected with, and potentially something that maybe holds much more of a virulence potential as it's released out. So the shift gives us more of an opportunity when we think about maybe pandemic sorts of situations, where it's really more of a novel player that becomes, the dominant strain, and we see it spreading, all right? So really depends, all right? And oftentimes we see a little of both kind of a play, some drift giving us some subtle variation, and then the potential is there for an individual to harbor both, right? Lending to something like this, right? There are other ideas. This truly is the last slide, but other ideas with different species and flus that they may have, all right? I was scrolling through the news before lecture earlier this morning, and I didn't click on it, but I saw some flu virus in cattle that's jumping over to people, and should we be concerned? Maybe we should, right? I didn't click on it because I feel like we should probably be concerned about anything that jumps from something to something, whatever. So that's maybe a concern. Bird flu is definitely something that we think of as a concern. And it really depends on some of these circumstances. The potential for a virus in a cow to jump to a human might be more likely than from a bird to a human. But from a bird to a pig, and from the pig to a human, that potential opens up all sorts of ideas about pandemic situations of bird flu and maybe other things as well, all right? And a lot of it has to do with the specific spikes and the host cell receptors that may lend to them being able to pull it in. And bird viruses don't really get into human cells very well, all right? But they can get into a pig cell, and pig viruses can get into human cells very easily. And that sets the stage for things potentially to jump ship, all right? All right. Questions or concerns? Farewell. Have a great day. Again, no class Friday. I will post that on Moodle. And the exam will get posted a little bit later, but it will open, like I said, Friday morning. All right. Have a good rest of the week. I will see you all Monday.